Today, we start learning one of the most fascinating and research-intensive disciplines in biology. Yes, right now, genetics, or the study of inheritance, and proteomics, the study of the proteins that are produced from the genetic code, is the hot topic in biological research, and for good reason. Now that we've mapped the entire human genome and the genetic codes of many other species, we are learning more about how life works at an astounding pace. For example, genetic and protein tests for colon and breast cancer are rapidly becoming developed and are being used to save lives because of the Human Genome Project. Yes, genetics is hot. We owe thanks for much of this progress to Brother Gregor Mendel, the father of modern genetics. In this Mendelian genetics subunit, we'll learn how Mendel used the scientific method to solve many of the riddles of heredity. Please review the SOLs covered in the Mendelian genetics subunit. Here are the objectives of Topic 1. At the end of this topic, you should be able to describe Mendel's experimental approach to investigating the question, how are inheritable traits passed from parent to offspring? Discuss the scientific merits of his experimental design, and discuss the outcome of his single-trait hybridization experiments. At the time of Mendel's experiments, which began in 1857, there were two hypotheses about how traits are passed from parents to offspring. The blending hypothesis is the idea that genetic material from the two parents blends together, like blue and yellow paint blend to make green. The particulate hypothesis is the idea that parents pass on discrete heritable units. Mendel sought to experimentally determine which hypothesis is supported by the evidence. Mendel designed experiments to answer the big question, how are traits passed from parents to offspring with garden peas? Just a few words about Brother Mendel, shown here with the other members of his monastery. He's the guy in the back admiring one of his plants. I guess he's the only one who brought a date. Born in 1822 in what is now the Czech Republic, he is generally recognized as an Austrian. As a boy, he was a peasant, very poor, and tended fruit trees for the lord of the manor. He entered the monastery in 1843, which sent him to the University of Vienna to train him to be a science teacher. He failed his teaching examinations 3 times and had a nervous breakdown in the process. Because of this, he wasn't allowed to teach college and eventually taught high school. Don't think that Mendel was a slacker. He probably just didn't test well. His life is a good lesson for those of us with test anxiety. Remember, tests are just one measure of knowledge. In spite of his lowly beginnings and academic problems, he was a brilliant scientist and a thoughtful and careful researcher. Mendel was very clever in setting up his experiment. He chose to use mathematics, and specifically statistics, to analyze his data. This approach had never been taken in breeding experiments. Then he selected a good experimental model, common garden peas, with which to test the prevailing hypothesis of inheritance. There are numerous advantages to using pea plants for genetic study. First, there are many varieties with distinct heritable features or characters, such as flower color, and character variants, such as purple or white flowers, that are called traits. Mating of plants can be controlled because each pea plant has sperm-producing organs called stamens and egg-producing organs called carpels. Cross-pollination, which is the fertilization between different plants, can be achieved by dusting one plant with pollen from another. In other words, he could carefully control mating experiments between different plants, or allow them to self-fertilize. Here is an overview of Mendel's experimental technique when crossing pea plants. Take a moment to study it. And let's look at these 5 steps in detail. Mendel used a four-step procedure to cross true breeding plants. True breeding plants produce the same trait consistently when allowed to self-pollinate. In this figure, you'll notice that the pea had both male and female sex organs. The stamens are the male parts, and the carpel the female parts. Let's say that Mendel wanted to cross a pea plant with purple flowers with a pea plant with white flowers. In step 1, he cut off just the male portion of the purple flower so it could not self-pollinate. In step 2, he took pollen from the white flower stamens, and in step 3, brushed it on the carpel of the purple flower. In step 4, pea seeds mature in the purple flower, containing sperm from the white flower and the egg of the purple flower. Thus, the pea seeds contain the traits from both parents. What would happen if Mendel didn't cut off the stamens from the purple flower first? I hope you said that the flower would self-pollinate, which would defeat the purpose of the cross. Let's review. Mendel used a four-step procedure to cross true breeding plants. True breeding plants produce the same trait consistently when allowed to self-pollinate. The purple flower pea plants were true breeding, and so were the white flower pea plants. He called those true breeding plants the pea, or parental, generation. He crossed the true breeding white flower plants with the true breeding purple flower plants and harvested and planted the seeds. Now for the results. In step 5, when the seeds grew, all the pea plants produced purple flowers. This generation is called the F1, or first filial generation. Filial is from the Latin word meaning child or offspring. Now let's think for a moment. What would you expect the first filial generation to look like if the blending hypothesis were true? I hope you said that you would expect them to be some intermediate shade between white and purple, such as lavender. This clearly was not the case. Mendel had gathered convincing evidence that did not support the blending hypothesis. But why did the F1 generation only have purple flowers? Where did the white go? We'll get to that question in a moment, but first let's review Mendel's experimental design. This is part of the beauty of Mendel's experiment. He chose to track the inheritance of characters that were easy to observe and were observable in an either-or manner. Purple or white flowers, dwarf or tall plants, smooth or wrinkled seeds. He also used true breeding varieties so that he could be sure that those observable traits were only coming from the parents. The only variation that would arise in his experiments would be the result of the crosses he made. I hope you can recognize that Mendel's experiments were well controlled and designed. They produced valid, repeatable results. Such sophisticated experimental design was not the norm in biology at the time, and therefore, most results were not repeatable or valid. Mendel's work demonstrates to us the importance of good experimental design when researching. Mendel usually conducted hybridization experiments, where two true breeding varieties of parents were mated. The true breeding parents are the P generation, and their offspring the F1 generation. He then allowed the F1 generation to self-pollinate, producing the F2, or second filial generation. Now we are going to answer the question, why were there no white flowers in the F1 generation? Having knocked down the blending hypothesis and supporting the particulate hypothesis through the F1 generation, Mendel wondered what would happen if F1 hybrid pea plants are allowed to self-pollinate. Which traits would appear in the F2 generation? Would the white flowers reappear? Now let's look at the whole experiment. As we have discussed, Mendel crossed true breeding plants for purple flowers with true breeding plants for white flowers. These plants are the P generation. As a result of this cross, all the F1 generation, the hybrids, had purple flowers. This refuted the blending hypothesis and supported the particulate hypothesis. Then he allowed many F1 hybrids to self-pollinate and observed and counted the flower color in the F2 generation. And look at the white flowers. They're back! We also discussed that Mendel was wise to collect numerical data and to use statistical analysis. In the F2 generation, he counted 705 purple flower plants and 224 white-flowered plants. Why did Mendel conduct almost 1,000 of these crosses? 705 plus 224 is about 1,000. I hope you said that he needed many observations to make sure that his analysis was accurate. Large data sets are more reliable than small data sets. In summary, when Mendel crossed contrasting true-breeding white and purple-flowered pea plants, all of the F1 hybrids were purple. When Mendel crossed the F1 hybrids, many of the F2 plants had purple flowers, but some had white. Mendel discovered a ratio of about 3:1 purple to white flowers in the F2 generation. In fact, he went on to carry out thousands of crosses over several years. Using the same experimental model as the white and purple flower experiments, he crossed many easily observable either-or traits, shown here. When analyzing his data, he saw a pattern emerge. A 3:1 ratio appeared in the F2 generation. For example, for every 3 purple-flowered plants in the F2 generation, there was 1 white-flowered plant. This observation held true for many other characters he tested, such as seed color, 3 yellow to 1 green, seed shape, 3 round to 1 wrinkled, and stem length, 3 tall to 1 dwarf. Mendel asked himself, why does this 3:1 pattern consistently appear? To answer that question, like any good scientist, Mendel analyzed and studied his data and drew some conclusions. Mendel reasoned that only the purple flower factor was affecting the flower color in the F1 hybrids. Mendel called the purple flower color a dominant trait and the white flower color a recessive trait. The dominant trait was masking or hiding the recessive trait. Mendel observed the same pattern of inheritance in 6 other pea plant characters, each represented by 2 traits. He knew that the heritable factors for white and purple flowers did not blend. Instead, the traits were carried separately in the eggs and in the sperm, and when united, those traits are still separate, or else the recessive trait would not reappear in the F2 generation. This alternative to the blending hypothesis became known as the law of segregation. Mendel didn't know exactly what mechanism in the plant was causing this pattern of segregation. In other words, he could explain what was happening, but could not explain why it was happening. We'll learn later that through many more years of research from other scientists, what Mendel was calling a heritable factor is what we now call a gene. That concludes topic 1 about Mendel's experiments. We'll practice doing these crosses in class. After that practice, and upon reflecting upon this lesson, you should be able to describe Mendel's experimental approach to investigating the question, how are inheritable traits passed from parent to offspring? You should be able to discuss the scientific merits of his experimental design and discuss the outcome of his monohybridization or single trait experiments.